Welcome to the Long COVID Coalition. My name is Leo Galland and I'm Head of Product Development at Medicinals Biotech. Together with Dr. Valentina Viduto from the Long COVID Foundation and Dr. Philip McMillan from VanElzakker, we are bringing the leading global experts on long COVID to the table in order to get a deeper understanding of the various multisystemic conditions at play. Collaboration in research, diagnostics and therapies are well underway for the benefit of the long hauler communities.
Hi there, I'm Mike VanElzakker. I'm a neuroscientist at Massachusetts General Hospital, and I use mostly neuroimaging and other techniques to try and understand long-term consequences from acute infectious illnesses.
Good morning, I am Carlo Brogna, and I am from Italy. I am a medical doctor and a researcher.
Hi, my name is Stephanie Seneff. I'm a senior research scientist at MIT, and I've been very interested in the issue of toxic chemicals and their exposure to toxic chemicals and human health.
I'm Dr. Shankara Chedi from South Africa. I'm a frontline doctor. I'm a general practitioner and a natural science biologist and been treating patients from the start of COVID. So I'm very interested in understanding the vaccines and, of course, long COVID and the mechanisms behind it.
Yes, welcome everybody. My name is Leo Galland and I'm Head of Product Development at Medicinals Biotech. I'm really looking forward to participate in this groundbreaking conference. See you all there.
Hi, my name is Dr. Valentina Viduto, and I am founder of Long COVID-19 Foundation. At the foundation, we aim to improve health and well-being of sufferers by contribution to long COVID diseases understanding and treatments. To do this, we work with organizations and individuals of medical professions, scientists, and long COVID advocacy groups to promote awareness of long-lasting symptoms, rehabilitation, and treatment challenges. This Congress will help us expand our knowledge base and seek for novel solutions addressing problems which will be discussed.
Thank you very much to everyone who is joining us on this journey with regards to long COVID. I'd like to take the opportunity to mention to you all about the vision and the idea of a long COVID coalition. And we really have to give thanks to Joachim Gerlach for using his expertise in terms of bringing people together to find solutions for this tremendously impactful disease. Now, what we have to do is that we are all still independent researchers. Say, for instance, I still do my independent research. I will still do my independent consulting with patients, and everyone else will continue to do that. However, there is an opportunity for everyone to pull together in a way that we can all work collaboratively to find solutions for patients with long COVID. And so with great pleasure, I'll bring in all of our panel and our speakers, and I'll ask them to very quickly introduce themselves and share a quick thought about what they are, and then we will start our presentation. Have I got everybody here, I think? Am I missing anybody? Because this is a full house. There is Manan. Okay, wonderful. So yes, so thank you everyone for joining us. This should be a tremendous opportunity for us to discuss some of these ideas. We've got 3 speakers and there are 6 panelists, including myself. Each speaker has about 20 minutes, slightly longer for Carla because of language right at the beginning. And each speaker will then have 2 panelists to discuss with them for about 15 to 20 minutes about the presentation. The purpose of the panelists is to try and simplify the concept so that everyone is able to come away from this presentation with a good understanding or a better understanding of long COVID. So the first thing that we will do is we'll just ask everyone to introduce themselves quickly, just in about 10 or 15 seconds. And so I'll start with Valentina, and just make sure you're unmuted, please.
Yes, hello, Dr. Valentina Vidota, and I am founder of Long COVID Foundation. Today I'm a panelist, and I will help you to learn on gut-brain axis and learn more about the bacteria. So looking forward to it.
Thank you. And Mike?
Hi there. I'm Mike VanElzakker. I'm a neuroscientist at Massachusetts General Hospital and Harvard Medical School. I'm going to talk a little bit about neuroimmune communication today.
Okay, thank you. Joachim?
Yeah, very big welcome from my side. I'm excited for the second conference we are holding today, and I'm a panelist today. I'm head of biotech. And we'll ask a lot of questions today and not speak much.
Hi, I'm Leo Galland. I'm a physician and an independent researcher in the US. I'll be speaking today about the gut-brain connection in COVID-19 with an emphasis on the gut microbiome.
Excellent, thank you.
Hi, I'm Dr. Shankara Chetty from South Africa. I'm a natural science biologist and a frontline worker. I've been looking at COVID from a clinical perspective from the start of this pandemic and hoping that we can get more insight into long COVID.
Excellent, thank you. And Stephanie?
My name is Stephanie Seneff. I'm a senior research scientist at MIT. I'm the author of this book, Toxic Legacy: How the Weed Killer Glyphosate is Destroying Our Health and the Environment. The book talks a lot about the gut microbiome and the interaction between the gut-brain axis. I'm a panelist, so I'll just be asking some questions today, but I'm looking forward to hearing the presentations.
Excellent, thank you. And we have Carlo.
Hi, I'm Carlo Brogna. I am a doctor in the research of Cranium Group, Italy. And today I will speak about the toxin and the SARS-CoV-2 and the long COVID condition.
Excellent. Thank you all very much. Where is Manan? I think I've Manan.
I'm Abdul Manan Beg. I'm the lead researcher of infectious disease group at Aga Khan University. Today I'm invited as a panelist in the second coalition conference. Thank you very much.
Excellent. Thank you all very much. Yes, so appreciate that from everyone. And so what we're going to do is we're going to go straight into our presentations, and we will start with Carlo.
Good evening and good morning, wherever you are in the world. So I am Carlo Brogna and a doctor in the research of Cranium Group, Italy. Cranium Group is a research facility, and we collaborate with other two facilities, one in Milan and one another in Naples. We also collaborate with the group of Professor Piscopo, Department of Biology, University of Naples. I thank the Long COVID Foundation Committee for inviting me. I am very honored and hope you will stimulated by my presentation. My presentation is based on toxin-like peptides, oligopeptides that we have been fortunate to observe in COVID-19 patients. I have noted that this issue has a lot of discussion in these past weeks. I want to clarify a few points today. The things you will hear do not deviate from the current knowledge, but it is, in my opinion, and it had an aspect that had not been considered yet. In the first part, we will review together some concepts on long COVID, neuronal receptor, and acetylcholine signaling. The middle part is focused on toxin-like peptides, what they are, what they came from, why they are important, how to recognize them, and the bacteriophage behaviors of SARS-CoV-2. In the last part, I will describe how this new observation can have clinical and industrial implications. I will not discuss or address this topic, vaccine, hydroxychloroquine, ivermectin, or the other product. This presentation reports only what we have tested in the laboratory and in our clinical practice. According to the WHO, the post-COVID-19 condition occurs in individuals with an history of probable or confirmed SARS-CoV-2 infection, usually 3 months after the onset of COVID-19. You can see the common symptoms in the right of these slides. And all these symptoms may also fluctuate or relapse over time. Here I describe 2 studies, an American study that observes in 100 not hospitalized patients, a good portion of the patients showing brain fog after 1 year. Moreover, the subject interviewed over the time exhibit an impaired quality of life and the cognitive and fatigue domains. On the right of the slide, you can see an Italian slide involving over 200 patients hospitalized for SARS-CoV-2. After 1 year, a good part present pulmonary problems and motor impairment. So in conclusion, it seems that there is an increased risk for hospitalized patients to develop a long COVID syndrome. Many authors have seen that in COVID-19 patients, there is an increase in coagulation status. This also is evident in other viruses, such as HIV, HCV, dengue virus, influenza virus, and Ebola. In this line, so the coagulation has a huge role. The Guillain-Barre Syndrome could be as a manifestation of long COVID condition. It is usually rapidly progressive but self-limiting inflammatory polyneuropathy. 25% of the patients with Guillain-Barre syndrome had recent Campylobacter jejuni infection. Double infection of Campylobacter and other coronavirus was also found in the past. And also with Zika virus or poliovirus was showing this syndrome. The post-acute sequelae of COVID-19 meet the diagnosis criteria of myalgic encephalomyelitis chronic fatigue syndrome. This syndrome begins with a viral infection. Immune dysregulation driven by SARS-CoV-2 may also promote the collective imbalance of the human body's microbial and viral ecosystem. Let's focus on the one symptoms particular, the hyposmia. Hyposmia is known as a prodromal symptom of many neurodegenerative diseases. It is involved with the cholinergic system, and also other coronavirus in the past was linked with the hyposmia. In this slide, we can see some conditions in which it's possible to observe hyposmia. Parkinson's disease, dementia, schizophrenia, myasthenia gravis, Alzheimer's disease, other coronavirus infection, poor industrial hygiene, and also snake bite. So there is a conflict in my mind, because from on the one hand, we can observe that the patients with hyposmia and the infection of SARS-CoV-2 ranking the symptoms from a few days to 3 weeks in many of the patients. On the other hand, the epithelium of olfactory bulb that should be affected by the viral replication takes 60-90 days to regenerate. So it is a flow in the neural transmission. Is it a flow in neural transmission, or it is a disruption of the olfactory epithelium by the virus. And this is important to show this slide to concentrate the importance of the autonomic nervous system. Few concepts. The autonomic nervous system regulates most of our biological function, breathing, circulation, and more other. It is divided in sympathetic, parasympathetic, and enteric system. Its activation occurs through the interaction of macromolecules and receptors. However, we must remember that many molecules can bind to the same receptor, and this link is variable over the time and depend on many factors. The most important receptors of this system are nicotinic and muscarinic receptors. So the effect can be prolonged when the toxin bind these receptors, and the toxin bind this receptor many times abnormally activate these functions. The drugs or the cholinergic medication that act on this system are divided in two types of drugs: the direct agents and the indirect agents. The toxins are classified as an indirect agents. And it is important to note that the indirect saturated agents inhibit two enzymes very important, that are the acetylcholinesterase and butyrylcholinesterase enzyme. They serve to break down acetylcholine. So when the level of acetylcholine increase dramatically, this enzyme decrease, and the dosage of these enzymes, as recommended Nakayama, is very important in COVID-19 patients as a prognostic factor. This is a slide in which we can view all symptoms that occurs in the cholinergic dysfunction, and many symptoms fall in the clinical picture of COVID-19 patients. Here we are in the middle of my presentation. This that you can see is the paper in which we describe the toxin-like peptides that we found in COVID-19 patients. You can see the list in the paper along with the mass spectrometry method used. Important thing to note is that each patient has over 17 peptides, toxin-like peptides. Each of them is repeated several times and with amino acidic variation. So we have considered a bacteria type of mechanism, because classically they are the ones who produce protein with frequent mutation. So by performing culture of the bacteria that came from the human microbiome of COVID-19 patients, we observe their genesis. In the imaginated model, SARS-CoV-2 does not pass unnoticed through the human microbiome before attaching the epithelial cell, but results in their disruption with the production of abnormal proteins that we have called toxin-like proteins, peptide. In this slide, at the center of this slide, you can see it is visible a conotoxin modified that we always found and that acting on acetylcholine receptor. This is a second paper closely related with the first, where we arranged an experimental model of 3 bacterial cultures. Simple A are the culture from COVID-19 positive patients. Simple B are cultured bacteria from healthy person but contaminated with the supernatant from simple A, and simple C was obtained from the only bacteria of the sample E and placed in a new culture. We observed an increase in viral RNA load from 0 to 30 days. At this point, we also performed the antibiogram to observe the viral load and observe that the viral load continued or not to increase with some antibiotics. And these 4 antibiotics on the right of the slide stop the viral RNA load. At this point, we investigated under microscope. We performed the electron microscope scan to better see the virus and bacteria interaction. In addition, we have seen if the proteins of the virus with the immunofluorescence microscope increase in our bacterial culture. And as you can see in this diagram, we report the increase of viral proteins after 30 days of culture. All internal control were made with mass spectrometry. At the fluorescence microscopy, you can observe in the merged image how gram-plus bacteria are linked with the signal of nucleocapsid protein of SARS-CoV-2. We compared new generation sequencing data before and after 30 days of bacterial culture. In the presence of SARS-CoV-2, some bacteria increase and others decrease. Therefore, we wanted to experiment with the bacterium of genus Doria, the formation of lytic plaque, with the supernatant containing the SARS-CoV-2. But what I consider the definitive experiment test was this one, in which we add the nitrogen isotope N15 in our bacterial culture of 30 days. And after another 7 days, we observe the presence in mass spectrometry of the isotope of nitrogen in viral proteins, nucleocapsid and spike proteins. So at this point, we have no more doubt. So SARS-CoV-2 has a bacteriophage behavior. The most interesting data is about bacterial toxin. Phospholipase A2 and conotoxin and other toxins that we have found, they continue to be produced by the bacteria behind 30 days. And also when the supernatant was removed with the SARS-CoV-2, the bacteria continue to produce them. But the novelty is that bacteria continue to be active, and so bacteria stop producing toxin very, very slowly. And at this point, we have tested the same antibiotics of the first experiment, and we observed that some antibiotics decrease their toxin release. The surprise was that amoxicillin and with clavulanic acid and rifaximin had the good efficacy. This suggests that the things are much more complex than we think. One class of bacteria may produce toxin, and another class of bacteria are involved in the bacteriophage mechanism. This slide shows our internal industrial data. It is an observational analysis of 100 COVID-19 patients who were symptomatic, not hospitalized, and treated with antibiotic by us. We noted that if the treatment is started immediately at the first symptoms, we have a clinical cure period of less than 7 days, compared to when it is delayed or if other drugs are combined or compared to use of the single antiviral. But then again, I can see that it's intuitive to think that if the toxins are present, we need to act immediately and not wait or not without waiting. So in this slide, I report two extreme examples of cured patients, but we have a large case of history. On the left, it is represented a long COVID patient that we have treated 10 months after the acute event. She still had hyposmia and abdominal pain, and after 7 days of rifaximin and probiotics, she regained her sense of smell and recovered. On the right of this slide, there is a case of a girl who was treated a month after the first positivity and where she had classic symptoms and complications from COVID-19, including neurological disorder, hyposmia, and dysgeusia. We started the first day with high dosage of amoxicillin with clavulanic acid. She recovered in 36 hours the sense of smell. We cut the dose, the second day, we cut the dose of an F, and hyposmia and other neurological symptoms reappeared. So we resumed the high dosage at day 4, and at day 10, the girl was clinically recovered. Summarize the important points. Accumulation of toxin. The accumulation of toxin saturates molecular receptor. In case of toxin infection, the time factor is crucial. The bacteria of microbiome represent the first line of defense. They produce toxin and partially replicate the various bacteriophage mechanism. The bacteria do not stop produce toxin even when the viral load is absent. The immune system must work against both viral proteins and toxin. The gut microbiome is a reservoir of this phenomenon. If we consider the bacteriophage mechanism and the toxin production by bacteria, the interaction mechanism of the virus and its proteins with the epithelial cell became much more complex. In addition, we activated mechanism of the immune system. The activated mechanism of the immune system also became much more articulated. So, how is the virus really presented from bacteria to the human cells? The clinical scenario that we might have are multiplied from symptomatic patients who are positive on molecular tests and have antibody titers to those who are negative on molecular tests and have no antibodies. So, how do we act? Regulate the toxin rate and viral replication. Acting on toxicological dilution. Corticosteroid only if necessary, because the immunity system must work against many proteins, viral and toxigenic, and so probiotics and antibiotics. The cytokine storm is much more complex because it is also actuated by the toxin. It is possible to cure the entire world with antibiotics? Of course not. The increase in antibiotic resistance bacteria must be considered. But above all, you must consider that the end point should be stop the toxin. The most powerful toxins that we have found are phospholipase A2 and conotoxin. And so the end point is stop to continuous production of the toxin, block them at the receptor level, maybe at the antidotes. Many studies, we are seeing many SARS-CoV-2 reinfection persons, and they seem to have a more attenuated clinic. The virus has a dual mechanism. It attacks the human cell, but also the bacteria in the microbiome. Some question: could the double or a dual vaccine solution be useful? A vaccine that trains the bacteria, such as an oral attenuated virus vaccine and an antitoxin vaccine? Is this a lockdown pandemic, or should it be accelerated? If you think about it, Dr. Sabin, with his polio vaccine, solved the problem by infecting everyone. This observation prompted us to review many other autoimmune diseases from another aspect, the toxin aspect. One day, maybe, we will be able to engineer bacteria, giving them resistance against this type of viruses, making them produce toxin and antitoxin, but also proteins that are indispensable for the human. Bacteria must be seen as a resource, not as a problem. At the end, many studies show us that the presence of the virus is in the stool for many days. And on the other hand, we observe the bacteriophage mechanism of the virus. So it's clear that there is the cycle of urban wastewater among the routes of spread. It is also clear that after this examination, the weather determinant is not the sun or the heat, but the frequency of rainfall and contamination of rainwater. How to intervene? It is necessary to decrease the bacterial load of the wastewater at this point. Thank you so much, and God bless everyone.
Thank you very much, Carlo. This is actually quite huge. And what I'm going to do is I'm just going to bring in our panel to be able to discuss some of what you have said. And before I step out and let you talk to the panel, here is what one person said. It says, you know, they are already considering you for a Nobel Prize, Carlo, because of this kind of work. So I'll let you get on the discussion. We have about 15 to 20 minutes with the panelists. So I'll let Joachim and Stephanie get on with that discussion.
Okay. Should I start off with a question?
Please do,
yes.
Stephanie.
Okay, yes, I want to say a fantastic talk. I'm so excited to see this role of the virus as a bacteriophage. Really fascinating, and the toxins produced by the bacteria causing the problems that linger. That's really awesome. My first question has to do with PLA2 and metalloproteinase, and I wonder they can also be produced outside of the gut in the bacteria in the gut. And it's elevated in the serum, and I'm just wondering whether what you see in the serum, is that due to the bacterial production, or could it be due to other production in the body of PLA2?
Oh, okay. Thank you for the question. Many research find phospholipase A2. So the phospholipase has a clear role in the pathogenesis of disease complication. We had focused on bacterial ones, but it is not excluded that phospholipase A2 or other toxin like metalloproteinase can be present from other production or cell production. So it is not to exclude, and many other authors found them.
Thank you. Do I just keep going, Joachim, and then you?
Sure, you go ahead.
Okay,
Yeah.
okay, great. Is the continued production of toxins, even after the disappearance of SARS-CoV-2, just an autopilot program in the bacteria? So does the general treatment with amoxicillin and rifaximin make sense in long COVID, or can probiotics alone do the job?
Soltanto allora la continua produzione delle tossine,
Sì.
oppure deve essere affrontata con gli altri due elementi, o può essere soltanto il probiotico che fa da solo?
Okay. No, in our opinion, not only the probiotics is necessary. The probiotics are is the most important drugs to assume, but in our opinion, also antibiotics is important in this to stop this toxin production. So, but I show in my last slide, it is not possible to treat all the world with antibiotics. So we need more other intervention or drugs to that acting on this point.
Thank you.
Thank you.
My third question, previous publications talked about PLA2, but you could also mention many other phospholipases besides PLA2. Or do we need to monitor all of them, or is PLA2 the important one, the primary one?
Okay. Yes, we found not only phospholipase A2, but also other type of bacterial phospholipase and phosphodiesterase. The phospholipase A2 is very important and has a key role in the active inflammatory process and downstream the arachidonic pathway, including the thromboxane pathway. Coagulation values will also be altered, including the dimer. So it is important also the other phospholipase, but I think that like our authors say, phospholipase A2 is one of the most important.
Thank you. And do you think this can be included in standard long COVID diagnoses?
I think that we must perform, or the industrial will perform the test that take in consideration the phospholipase A2. Maybe the finger test that can evaluate the presence of the phospholipase A2 or the other toxin, or maybe a finger test that can give us the indication of acetylcholinesterase concentration if it is low or high.
Thank you. Is there a treatment that mitigates the effects of the conotoxins?
Can you repeat, please?
Is there a treatment that mitigates the effects of the
mitigation of conotoxin? No, conotoxin is a very particular toxin. So now we know that it is produced by bacteria and we can act on bacteria. But it is very necessary for conotoxin maybe to create an antidote.
Okay, I'll jump in.
Okay, you want to go in now? Yes.
Thank you, Carlo, for your groundbreaking presentation. And I think you brought a big chunk of the knowledge now to the table of long COVID condition that was overlooked and not. And I'm very happy that we had a chance to contact you and go through that. So I have some more questions. Looking at your pictures from the microscopic slides, do you know how does the SARS-CoV-2 enter into the bacteria? What is the entry mechanism? Is there any idea on what the receptors are, for example, and the mechanisms?
Okay, yes. This topic is under study in our research facility, but we have appreciated the work of Dr. Ibrahimi and colleague that have performed some studies on interaction of a possible interaction. They are bioinformatic study of a possible interaction of spike protein and protein of bacteria that are similar to receptor ACD2. So we need more study this argument and this topic.
Yeah, then,
See,
that is
we need to study. We have no idea in this moment. It is a phenomenon that we observe, but we must to study well this argument.
Then the next question, the virus itself is spreading toxins that looked like on your slides. When I saw that around the coronavirus, you had toxic kind of particles. Can the virus itself create the toxins, or are they being produced by the bacteria?
Like we show in the slide 23, we observed the toxin also when the virus was removed from the bacterial culture.
Mm-hmm.
So it is, I think, but we must study better. I think that is an old mechanism of bacteria, a self-mechanism of immunity of bacteria. But because we observe in the vitro, but then in the practice, that the toxin were produced also without the RNA load present in the bacterial culture. So maybe their genesis is a mistake of the genomic sequencing, but we don't know in this moment. We must perform more experiments.
Interesting. If you need any help, just let us know. The other question was, like, if you look at the bacteria that is infected with the virus, can then the bacteria with the virus itself replicate so that we can observe maybe a growth of bacteria culture and virus at the same time? Is that happening?
Yes, in our culture, we observe the proteins increase of the virus and also the toxin increase of the bacteria, the toxin increase. So, and in another culture with other two bacteria, the Doria genus, we have performed the two-plaque assay with Doria genus and the Faecalibacterium pasteurinae, that and in this bacteria we observe a lytic phenomenon. So in our study, there are some bacteria that increase and other bacteria that decrease. And with some bacteria, the virus increase, and with other bacteria, the virus decrease. So we think that the virus in this moment have a double mechanism, lytic and lysogenic from the together side, and this is maybe the condition that give more complication.
Yeah, this is really horrific if you look at all the different kind of mechanisms, how that plays out. And now, last question from my side. We all like to give probiotics or take probiotics. And if we would give now a probiotic, like a bifidobacterium or any bacterium in which the virus is replicating, could that be also like a detrimental action? Because then we give the virus food for replication.
Okay. Thank you for the question. So this question I can answer only with my practice, not with the test laboratory. In the practice, when we give to the patients antibiotics, amoxicillin and rifaximin in association, the major patients restore their symptoms in 5-6 days. But we give also the Lactobacillus reuteri and Bacillus clausi, and they work well together, the antibiotics. So, but this is in our practice, come si dice, consiglio, advice. But we have not tested in laboratory if the virus attaches also the probiotics.
Thank you so much, Carlo, for your groundbreaking work and these insights. We still have a lot more questions, I think, and we could pass the whole day talking, but there's other speakers. So Thank you very much for this.
Thank you, everyone.
Findings and presentation of yours.
Thank you, everyone.
Excellent. And yes, Carlo, I myself will have some questions for you, but I will leave that for the open discussion. But we will shift on to our next speaker. So thank you again very much, Carlo, and stay with us, and we will certainly be discussing further with you a little bit later on. So moving right along to what is quite a fascinating area of research with regards to long COVID, we have got Mike VanElzakker.
Yes, yeah. So yeah, I appreciate the invite, everyone. So I'm going to talk about sickness and the brain, the neuroscience of how we feel infections and inflammation. It's sort of going to be a basic neuroscience, neuroimmunology talk, sort of broad and mechanistic and anatomical, just explaining some of the reasons why people feel the subjective feelings that they feel when they're sick. And I don't have anything to declare, and my presence isn't an endorsement of anyone else's statements. I actually have COVID right now. I tested positive, I guess, 3 days ago, so I feel pretty sick. I'm going to try and get through this. I'm triple vaxxed, but I had a breakthrough infection like so many others are having. You know, we all know about long COVID at this point. It's something that has really, you know, been a topic in general media and around the world, our friends and loved ones. It's a serious issue. And it's something that we're going to have to really dig in and understand. So I thought that talking a little bit about the basic neuroscience of sickness and illness might be helpful here. This is a study from Hannah Davis and her collaborators. It's a group of essentially patient scientists and patient advocates, I suppose, where they followed people with long COVID early on and talked about the symptoms that people were experiencing 7 months after their onset. And the symptoms here are sort of centered around fatigue, brain fog, essentially the feelings of being, the very feeling of being sick, the sickness feelings that we all have, whether it's a flu or a strep throat, bacteria or virus, the feeling of being sick. Autonomic problems are really common. Some recent studies showing about 2/3 of people with, you know, the long-term consequences of COVID showing autonomic problems and pain. So I'm going to stay focused on those categories of symptoms: sickness, autonomic problems, and pain. Sickness, autonomic problems, and pain. And we'll talk a little bit about those. You know, many of us have been working in the world of myalgic encephalomyelitis for many years. You know, this is a phenomenon we've seen before, in which an acute infection, there isn't a full recovery, and people, you know, feel sick sometimes for many months or many years afterwards. And, you know, early on, we sort of knew that this was going to be, you know, in a minority of people, but still a significant minority in people that deserve healthcare. So if we look at the symptoms of myalgic encephalomyelitis, it's very similar to those symptoms that I just talked about in the long-term consequences of COVID. Now, with COVID, there's going to be some people who have lung damage, tissue damage. They were in the ICU and had a rough intubation. And those are particular types of insults, problems that aren't quite the same thing as ME/CFS, but there's going to be an overlap with ME/CFS. And again, if we look at the symptoms of ME/CFS, without going through them all, this is the international consensus criteria. It's striking that they're also focused on sickness, autonomic problems, and pain. Sickness, autonomic problems, and pain again. If we look back at when the term myalgic encephalomyelitis was first coined in a 1956 Lancet article, in which the clinician at the time wrote into the Lancet saying, look, we've had these outbreaks. If you look here in the middle column here where it says virus, some of the times we knew what virus was causing the outbreaks, and some of the times we didn't. You know, this is before PCR was invented. And yet it was causing this long-term problem. The term benign here just meant that it wasn't killing people like polio sometimes did, but it wasn't meant to say that it was minor, just that it wasn't killing people. So what you can see here basically is that several different kinds of virus, to the extent that they knew, were causing these same consequences, or apparently similar viruses are causing the same consequences. It's worth noting too that there's other the word post in big quotes, I think, post-Ebola syndrome here, for example, where people have an acute Ebola, infection, they survive, and then they go on to have symptoms that include musculoskeletal pain, headache, in this case, ocular problems, but centered on those same symptoms. Post-Zika, a similar virus to SARS-CoV-2, again, causing somewhat similar symptoms. And here, the SARS-CoV-1 causing relatively similar symptoms, again, sort of focused on sickness, autonomic problems, and pain. People just describing these same symptoms. So we wrote about this a little bit in a review article early on, trying to make the world understand, for one thing, that this isn't quite as mysterious, I think, as we sometimes hear. We do have some understanding of what the mechanisms are. We do have some understanding from, for example, post-Ebola, quote unquote, that in that case, the Ebola virus was still around. When they finally tested in sanctuaries, they tested semen, and even in the eyes, there was live viable virus in those cases. And there's some accruing evidence that the same may be true in so-called post-COVID. So given that I named a bunch of different viruses, how can these disparate problems drive similar symptoms? How can we end up with the same sickness, autonomic problems, and pain from different causes? Well, I'd like to focus in on an area of the brain called the brainstem. And here's a scan of my brain at 7 Tesla. And I'm focusing in on the brainstem here. If we blow it up and Photoshop out the vasculature, it looks like this. And here's the anatomy. It's an area where cerebral spinal fluid flows through very thin aqueducts nearby. And I'm going to blow it up even further so that we can sort of focus on these anatomical nuclei within the dorsal brainstem of the brain, because I think it's important for the symptoms that we see and a mechanism for why we may see these symptoms. There are a bunch of structures and circuits that are in really close proximity, and they overlap and crosstalk. They communicate with one another, and they probably drive some of this, the subjective feelings that we feel when we're sick. So here, sort of centered, that I've highlighted in red now, is the nucleus of the solitary tract, NTS, nucleus tractus solitarius. And this is a place where the vagus nerve enters the brain from the body. Okay, so the vagus nerve is the 10th cranial nerve. I'll talk about that in a little bit. It shares the same Latin root as the word vagabond, right, meaning to wander, which means that it wanders all over the trunk, all over the torso. It innervates the lungs, the bronchia, the gut. If there's inflammation or infection happening in the lungs, if there's a dysbiosis in the gut, the reason that the brain knows about that is because the vagus nerve detects it. And this is where the vagus nerve enters the brain from the body. It's where a majority of these afferent or sensory vagus nerve cell bodies live. So nearby the nucleus of the solitary tract is an area called the area postrema. So this is a circumventricular organ without a complete blood-brain barrier. In other words, there are some areas of the brain where there's what we call a fenestration, or a window in the blood-brain barrier, which is sort of a barrier to prevent the blood from having easy access to the tissue. This is an area where there's windows in that so that the brain can sort of sample larger molecules. It's really important for vomiting and nausea. A lot of patients have ongoing problems with nausea with these complex chronic illnesses. These areas here are both body to brain and brain to body. So the two areas I talked about represented the body communicating to the brain, but now I'll talk about the brain communicating back to the body. So the dorsal motor vagus is the vagus nerve going in the other direction. So it's an autonomic nuclei. It's the site of parasympathetic cell bodies, or soma. And it's the main source of how the vagus nerve innervates the trunk organ. So the vagus nerve has control over, for example, heart rate. So if there's a problem with tachycardia, high, fast heart rate, even going from lying down to standing up, that may be an important part of why that's happening, because of this structure here. The dorsal reticular nucleus down here in red is important for inflammatory pain transmission. Now notice what I've talked about so far are sickness, autonomic problems, and pain, right? So the very feeling of being sick is the nucleus of the solitary tract, which I'll talk about in a little bit. The autonomic problems are represented at least partially by the dorsal motor vagus. For example, the tachycardia, fast heart rate. And here's a nucleus that's really important for the transmission of pain. So if there's a situation where we call hyperalgesia, in other words, where something that would normally only hurt a little bit ends up hurting a lot, we call that hyperalgesia. Or something that shouldn't hurt at all ends up hurting, that's called allodynia. And that may be important in this structure here. The ventrilateral medulla is involved in autonomic control of blood pressure and breathing. So again, autonomic problems, to the extent to which they exist in patients, may be sort of centered on these nuclei right around here. And the nucleus ambiguus right here is, again, an area that's important for controlling heart rate, which we know a lot of people with so-called long COVID or PASC have problems with tachycardia when they go from lying down to standing up, for example. Some people qualify for the POTS diagnosis, not all, but some people just have autonomic problems. So, I talked a little bit about the vagus nerve, and I'll talk a little bit more about that here. So again, it wanders all over the body. I've drawn a little diagram here from an older paper showing the different ganglia and paraganglia that are all over the body, really, all over the trunk. The vagus nerve innervates the bronchus, the lungs, very densely. So there's a lot of vagus nerve terminals at the lungs. So this is really important because inflammatory molecules like cytokines, you've all heard of cytokine storm, right? The virus obviously triggers cytokines. Well, cytokines are relatively large protein molecules that don't easily cross the blood-brain barrier. So they don't just flow into the brain like a steroid hormone does. Well, how then does the body know that there's cytokines? Well, it knows because of the vagus nerve. There's chemoreceptors on the vagus nerve that detect the presence of those cytokines, and it sends a signal up to the brain that says, hey, guess what? You've got inflammation. You're sick. And that is the reason that we feel sick. It's the body's behavioral response to inflammation saying, you need to preserve your energy, go to bed. You shouldn't be spending your energy on jogging or other pursuits right now. You need to go to bed. So interestingly, for example, if you inject inflammation into a rat, the rat will not run on a wheel anymore. They won't eat as much, etc., unless you've cut the vagus nerve. And then the rat will act normally, even though you have injected inflammation, because the brain doesn't know that the body is sick. So one of the things that the vagus nerve does is to detect peripheral catecholamines. So, for example, epinephrine or adrenaline. You've heard of an adrenaline rush that's produced, right on top of your kidneys, the adrenal on top of kidneys. Well, again, that doesn't actually flow into your brain very easily. It's the vagus nerve that detects those chemicals and signals to the brain. And it also detects local immune signaling molecules in the way that I've just described. So cytokines, inflammatory proteins are detected by the vagus nerve. Now, importantly here, because the vagus nerve wanders all over the trunk, the inflammation can be local. So you could have a gut dysbiosis, for example, inflammation in the gut. And it doesn't even have to be inflammation flowing through the entire circulatory system. It doesn't have to spill over into the blood, for example. It can be local. And because the vagus nerve has so many branches, it will detect that and that will drive the feeling of being sick. So it surveys the gut microbiome. That's a major job. And it initiates the central illness response, which includes, as you might guess, the feeling of being sick, autonomic problems, and pain. So these ongoing symptoms that people may feel from different forms of illness. And we can measure that with PET scanning, brain scanning, which is one of the things that my lab is doing right now, is studying this in so-called long COVID, measuring inflammation in the brain. So when there's immune cells in the periphery, here I've drawn a monocyte and a macrophage, they don't easily cross the blood-brain barrier. So I want to talk a little bit about how the signaling happens. When these immune cells detect either pathogens, that's a virus, a bacteria, they activate. They sort of turn on, and then they produce cytokines. Now, one way that they could affect the brain is if they flowed right into the brain through the blood like a hormone does. Now, that doesn't really happen all that much. It happens a little, but not all that much. So another way that it happens is crossing the circumventricular organs. There's about a half a dozen of those circumventricular organs, including the area postrema, which I had mentioned before. So the cytokines, inflammatory molecules, can flow across those circumventricular organs and affect the brain and cause the feeling of being sick. Or interestingly, the immune cells themselves can be taken across the circumventricular organ, and they can enter the brain. This is actually a way that some pathogens have evolved to have a Trojan horse mechanism for infecting the brain. They infect the immune cells, and then the immune cells are brought into the brain. But another way that these cytokines can affect the brain is when they're detected by the vagus nerve. It sends, and because the vagus nerve is all over the place, it can be local, and it sends a signal up into the brain that triggers an inflammatory response on the brain side of the blood-brain barrier. So it allows local immune signaling to have a broad effect. It doesn't have to be infectious. It can also be injury. So, for example, if there's lung damage from the virus, even if theoretically the virus was fully cleared, you could have ongoing inflammation just from the tissue damage. And it triggers this mirror response of glial activation on the brain side of the blood-brain barrier. And this is something that they've measured in chronic fatigue syndrome in some studies, not others, and that we're studying in long COVID in my lab. I wanted to talk just for a minute about those glial cells. Glia means glue in Greek. It used to be thought that it was just holding neurons together. This is microglia of the brain. So when they detect pathogens or inflammation, they change their shape, they activate, and they produce, they actually change into this sort of different form, and they produce a bunch of things that excite neurons. So they produce cytokines, which I've represented with the little triangles, and they also produce things that excite neurons, which I've represented by these little lightning bolts. Things like glutamate, bradykinins, prostaglandins, ATP, nitric oxide, lots of different things that excite neurons, and a protein called the translocator protein, which we can measure with brain scanning. After that happens, the glia enter this state called priming. They're primed. What that means is that they're sort of hypervigilant, I suppose you could say. And if a second immune response comes and affects those glia, then the second response is even bigger. That's what I'm showing here. The second response is even bigger. So one of the things that I think can happen is that an initial inflammatory response from an initial infection, say the SARS-CoV-2 infection, can prime the glia, and then the glia becomes sensitive to things like gut dysbiosis. So someone that might have a little problem with gut, if they don't have this problem, maybe they won't even be affected. But if they have primed glia, then maybe they will be affected. And that can drive sort of brain fog symptoms. You know, I talked a little bit about that area in the brainstem, the nucleus of the solitary tract. If there's glia that are excited there and they're turning on neurons nearby, well, look at all the nuclei that I talked about that were nearby that control sickness, autonomic problems, and pain. So it's almost like having a little amplifier on these nuclei. Now, that's not to say that we should immunosuppress these patients. I'm not sure that that's the right call. What it means is if there's something driving inflammation, whether it's a gut dysbiosis, whether it's other infectious, whether it's injury, whether there's viral reservoirs, that those things need to be addressed because there's a sensitized system in people that make them extra sensitive to insults, including things like toxins and infections. I've talked a little bit about this in a previous paper that you may be interested in learning more about neuroinflammation. You can look at it here. And I'd just like to thank my collaborators that helped me with all the brain scan projects that we have going, including in long COVID. Thank you very much.
Excellent. Thank you very much, Mike. I really appreciate that. And again, I'm seeing so many parallels with all of the presentations and seeing how they all interact and come together. Again, I'll have to save my questions for later, but I'll bring in here your panel. We've got Shankar and we've got Manan here, and I'll leave us here for about 15 minutes or so, and they can ask you some questions, and then I'll come in and we'll move on to our next presenter.
Hi, Mananda, would you like to go first?
Yeah, sure. Okay. So, Mike, thanks for a very nice presentation. Okay, this is my second interaction listening to you. Last time I had that fascinating CSF flow thing observed, okay, that we didn't see today, but then I was, first of all, let me begin by thanking the Long COVID Foundation and Valentina to bring all of us together, you know, because these type of efforts are the ones, okay, which are needed, and we should always acknowledge the heavy contribution from the Long COVID Foundation that actually brings us together. My question to you, Mike, is that nowadays, okay, you must be hearing a trend of vagus nerve stimulation, okay, in long COVID. My question is that, as you described, you know, the cytokines, they irritate the vagal nerve ending, the vagus goes, and it relays in those centers that you mentioned. My question is that if we do a vagal stimulation, okay, and it actually would transmit signals to the brain, how would it be helpful in any COVID, I mean long COVID or the symptom? At the end of the day, it's a stimulation, be it cytokine or electrical stimulation. So I want your thoughts on this, please.
Sure. Yeah. Thank you very much. And it's nice to see you again, for sure. So yes, vagus nerve stimulation is something that people have talked about. I think it's worth pursuing and worth doing. You know, we're trying to get a study going of that to see. Now, I think it's important to note with vagus nerve stimulation that if there's, for example, if there's viral reservoirs, for example, it won't address that problem, right? It doesn't necessarily get to the root cause in that circumstance, but I think it can be helpful. You know, if there was a clearance, for example. The vagus nerve is really interesting because it goes in two directions. So from the body to the brain, the afferent or sensory direction detects inflammation and sends a signal to the body, from the body to the brain that says you're sick, feel sick, go to bed, etc. In the other direction, there's what's called an anti-inflammatory reflex. So in the efferent or motor direction of the vagus nerve, there's a synapse in the spleen that ends up producing anti-inflammatory cytokines. There's different types of cytokines, right? So the thinking is that with a vagus nerve stimulating device, you could theoretically be sort of regulating the afferent direction, a little bit like a pacemaker. I don't want to stretch the analogy, but a little bit like a pacemaker in the heart, where it's sort of, you know, trying to regulate what might be a spastic or ongoing signaling a little bit. And then also at the same time, hopefully exercising that motor or efferent direction in the anti-inflammatory reflex. So there's pretty good evidence, for example, in sepsis, where that's a really extreme example of inflammation, right? A genuine cytokine storm that can be deadly, probably much more than what people with long COVID are facing. But even with sepsis, it increases survival times, probably because it's regulating both directions of the vagus nerve. So yeah, I mean, I think it's worth noting, and you know, most of the you know, I don't have any relationship with any of these companies. I talk to them, but I don't have any investments or anything. You know, most of the vagus nerve stimulating devices that I think I are most comfortable with tend to focus on the ear. There's what's called the auricular branch of the vagus nerve. It's the place of the vagus nerve that comes closest to the skin of the ear, and they stimulate right up here in the cymboconcha, or the tragus of the ear. They tend to use the right side. They tend to use the left side of the vagus nerve, so that it doesn't affect the heart. But yeah, vagus nerve stimulation is something that I think is really worth investigating, especially the ones that stimulate near the ear.
One more thing, okay, a follow-up question to your presenting slides, okay, one of which showed that blood-brain barrier thing. My question is that with the COVID infection, you know, and when I say COVID infection, I mean active COVID infection, wouldn't the blood-brain barrier be already breached by the SARS-CoV-2, okay, as it passes through cerebral circulation, slowly interacts with the endothelium? So if it's already breached it, you know, those cytokines that you were mentioning, okay, they are in the blood, they are just like like an easy track to go to the brain. So what are your thoughts that initial SARS-CoV-2 infection can affect the blood-brain barrier very initially in the stage of the infection, and then the cytokines have got a free pass to cross across?
Yeah, I mean, you know, the SARS-CoV-2 virus is capable of neurotropism. We know that. Whether or not that's what's causing most deaths, I think, is probably you know, maybe not so clear, or less likely, but you know, it clearly is capable. And the circumventricular organs, the choroid plexus, for example, are unfortunately a place where the virus can infect and have an effect. And the choroid plexus they're circumventricular organs that are the place where cerebrospinal fluid is produced. And this is an example where the virus, to the extent that it can get into blood, can end up interacting with the brain and penetrating into brain. And yes, you'd have two problems there. One is that the blood-brain barrier would be disrupted or enlarged in that circumstances. And then the actual just cells right there would be producing cytokines as well, right, locally, which would have a profound effect on the brain as well. So yeah, it's sort of bad news if that happens, I think. And blood-brain barrier is something that I think is an area of research that's really important to understand long-term consequences. The brain is very demanding of oxygen. It uses a lot of vasculature.
Okay. So very quick, last question, you know. We know that responses like fever, you know, actually we evolved it evolutionarily. They help us, okay, by providing us rest. My question is that if a cytokine transmits impulses via vagus to the brain, and it alters our behavior, that it wants us to rest more, isn't it a defensive thing? I mean, removing it, do you think is a good choice, or you think that this evolutionary thing should be kept upon for some time? Because it's a thing that we developed as a defensive mechanism.
Yeah, I mean, I agree with that. I think it's a tough thing for people to understand, is that something that is subjectively bad, like pain, is there for a reason, right? It's there as a defense. So is sickness. The very feeling of being sick is evolutionarily preserved for, you know, hundreds of millions of years, because it is a signal. It's the behavioral aspect of the signal. Go to bed, you're sick. Fever is a good example where it's part of the innate immune response. If a pathogen has evolved to replicate in a host, it's probably evolved to replicate it at 98.6 Fahrenheit or 37 Celsius. So raising the temperature makes it a little bit harder. You know, cytokine to brain signaling through vagus does mediate fever as well. So yeah, I mean, I don't think you want, you know, this is sort of my problem with immunosuppression in general. At some point, you have to immunosuppress. If someone's going to die of a cytokine storm, then you have to save their life. But I'm not so sure that that's the right call with the sort of chronic lingering symptoms, because it will make people feel better, which is the tantalizing thing. because feeling bad is part of the immune response, but it may not address the actual root causes. And those ought to be addressed better if we're going to actually get people better instead of sort of temporarily making them feel a little bit better.
Thanks, Mike, okay, for your great presentation. Okay, really enjoyed it. Dr. Shankara?
Thank you, Dr. Baig. Dr. Elzakker, I got a few questions. We've seen that Omicron has a propensity for neurotoxicity. I've seen a lot of patients presenting with neural symptoms, burning of hands and feet. There seems to be a spinal nerve root involvement in some, where they developed a sciatic kind of pain, those kind of presenting symptoms. And it ranges to a sudden onset of from patients becoming very depressed to mild psychosis and psychiatric issues. And we've established that we have a cytokine-like or hyperimmune response after about a week of initial viral infection. Now, the cytokine release is seen to be the trigger for these peripheral neuropathic kind of symptoms. And these kind of patients respond well to suppressing those cytokines, and it tends to suppress the pain, being the root of the problem itself. But with vagal nerve stimulation, causing the sick feeling, which is usually a physiologic process in trying to help you recover, is it your contention that this mechanism is because of prolonged inflammation somewhere else in the body, that the continual vagal nerve stimulation is causing the prolonged feeling of being unwell?
Right. So that's a good series of questions there. So obviously an ongoing inflammatory signaling will cause the sensation of being sick. That is sort of a straightforward possibility. But because of the phenomenon of glial priming that I mentioned before, that can be quite oversensitized. So glial cells in the central nervous system are relatively long-lived cells. So they can live for something like 8 years, some different kinds. They're replaced by their own replication. They're not replaced by circulating stem cells. And so there's probably some phenotype preservation across time. So in other words, if the glia become primed from an initial inflammatory insult, they're probably going to stay primed for a long time. And so I think that one of the things that can happen is if someone has a primed glia, that what normally most people could handle in terms of gut dysbiosis or ongoing problems probably will be a little bit sensitive in those folks with a primed system. So I think that is an example of where the ongoing signaling from body to brain through vagus doesn't really have to be all that profound, I would suspect. So that's one of the things that I think. And in terms of the neuropathy, it is true that there's an inflammatory component to that. Neuropathy means, for people listening, there's nerves way out near the skin that are important for pain. They're called C fibers. They're unmyelinated nerves. And, there's evidence from Anne-Louise Oaklander's lab, for example, and Peter Novak, two of our colleagues here at Harvard Med, that the C fibers way out at the skin, the intersection of the dermis and epidermis, become damaged and start to retract. Almost certainly inflammation is part of it. There may be other mechanisms that we're looking into as well that I think could be interesting. That may be part of why there's part of the diffuse pain that people can feel, sort of soreness and things like that. Unfortunately, that can happen way out at the skin or anywhere along the pathway all the way up. The increased problems can be caused. So the pain pathway goes through the dorsal horn, the dorsal horn of the spinal column. There's some evidence for inflammatory signaling there as well. So if there's ongoing inflammation, glial activation, maybe viral reservoirs from this or other viruses that are, 90% of humans have herpes viruses in our body. They like nerves. If there's herpes there, that could be causing inflammation. So yeah, I think that the pain and neuropathy can be caused way out at the level of the skin it can be caused at the level of the spinal cord, and can be caused in like an amplifier at the level of the dorsal reticular nucleus or anywhere along the way, or the periaqueductal gray, which is another area of the brainstem that's involved in signaling. So if the glia are acting like amplifiers, turning on nerves, turning on neurons, then that can enhance signaling to cause hyperalgesia. Something that wouldn't normally hurt can end up feeling painful. I hope that's a helpful answer to your question.
Secondly, Carlos mentioned the production of certain neurotoxins, conotoxins, PLAs. These might have more direct effect on acetylcholine receptors, the nicotinic and the muscarinic. Will there be an interplay in neurologic illness with long COVID between the exposure to these directly acting toxins and, of course, the global inflammation they can cause and the gut dysbiosis we see? And so we'll have a mix of different mechanisms causing the neuropathies that we see, both central and peripheral.
So if there are some sort of metabolite or mediators that affect acetylcholine signaling, that would, of course, have profound effects on vagus nerve. When acetylcholine was first discovered by a German physiologist, 100 plus years ago, he first named it Vagus-Stoff because it was the stuff of the vagus nerve. So acetylcholine is a really profound neurotransmitter for vagus nerve signaling, and is especially in its sort of parasympathetic output, control over heart rate and things like that. So if there's something that's directly affecting acetylcholine, some toxin or metabolite, then that would really, I think, profoundly affect the function of this system in a way that you'd have to address in order to get people better.
Thank you. Thank you for a great presentation.
Thanks for the question, guys.
Thank you very much for everyone here. And again, we'll ask you all to hold on, but we're going straight away into our next speaker. And I'll take you gentlemen kindly out of the screen to Dr. Leo Galland from the USA. So Leo, I have the pleasure of being one of your panelists. So I look forward to your presentation, and I'm sure I'll have a number of questions for you.
Okay. First, I'd like to thank Valentina, Joachim, and you, Philip, for inviting me to present at this conference. I'm going to be talking about the role of the gut-brain axis, and in particular the microbiome, in long COVID. Now, that's the way we do it. Okay. I will start with an overview of the gut microbiome and its systemic effects. Look at the way that these effects impact brain function and repair, the impact of COVID-19 on the gut microbiome, and the way that the gut microbiome may impact the course of COVID-19 and the risk of long COVID. And then consider the gut as a reservoir for persistent SARS-CoV-2 infection. I'll be looking at research that's been done by many different people, and suggest some of the strategies that I've advocated for therapeutic modulation of the gut microbiome to either prevent or reverse long COVID. Now, the big surprise of the Human Genome Project was the relatively small size of the human genome. We have about 25,000 functioning genes, despite tremendous complexity and diversity of our species. In contrast, rice shows relatively little and is the fruit of this particular plant, Oryza sativa, has almost twice as many genes that have evolved over 15 million years. So how do we as humans manage to do so much with so little? There are many scientists who believe that the answer to that is the human microbiome, which contains about 4 million genes. Now, genes are the templates for creating proteins, and there are two major types of proteins that are produced. structural proteins, and enzymes. And enzymes are catalysts of biochemical reactions. The gut microbiome basically represents the body's largest and most diverse ecosystem. There are about 100 trillion microbes there, 95% of the total, and there are more microbial cells in the gut than there are human cells in our body. Understanding the microbiome really requires answering two basic questions. Who's there and what do they do? Well, what they do is they produce a tremendous number of proteins, either structural proteins or enzymes, and those enzymes alter, create, destroy, and recycle. They do it to nutrients, they do it to toxins, to hormones, to human secretions. They may make nutrients more or less bioavailable. The structural proteins in particular stimulate the immune responses. Both the proteins and the structural proteins may modify systemic metabolism. They definitely alter gut immunity. They compete with pathogens. They activate the enteric nervous system, which connects directly with the brain. Much of that connection is through the vagus nerve, but not all of it. And they influence protein function in brain and muscle. Now, the structural components of the gut microbiome are recognized by the innate immune system's pattern recognition receptors. Microbial metabolism of host secretions and ingested substances yields products that impact human cellular function. Over 90% of the chemicals circulating in your blood originate with your gut microbiome. We know this from looking at germ-free animals who have no microbiome, and they have a tiny fraction of the number of chemicals that are found in circulating in blood compared to wild-type animals. Now, some of these substances are neurotoxins. Two that have been well studied are ammonia and D-lactic acid. D-lactic acid is not produced by human cells, but only by bacterial cells. Some of these substances may trigger systemic inflammation, which then impacts the brain through some of the mechanisms that Mike described. In particular, what's been studied are LPS, or lipopolysaccharides, which are part of the cell wall of gram-negative bacteria, and peptidoglycans, which are part of the cell wall of gram-positive bacteria. Some of these substances are directly and actively anti-inflammatory and stimulate tissue repair. And probably the most important and certainly the most studied of these is a short-chain fatty acid called butyrate, or butyric acid. Now, in the brain, and butyrate being a short-chain fatty acid, passes readily through membranes. It goes from the colon, where it nourishes the colonic lining and supports energy production, directly into the body, and it gets readily into the brain. And in the brain, butyrate stimulates the synthesis of a substance called brain-derived neurotrophic factor, at least in laboratory animals, which is really an important mechanism that the brain uses for repair. Now, there's some substances that are produced that may be helpful or harmful depending on their concentration and other factors. One of these is hydrogen sulfide, a gas. Hydrogen sulfide in the brain actually is neuroprotective, but in the gut causes inflammation, although it may depend on whether you're dealing with the stomach or the colon. And then there's a short-chain fatty acid similar to butyrate called propionate, or propionic acid. And although many benefits of propionic acid on peripheral metabolism have been demonstrated, in the brain, propionic acid may contribute to autistic syndrome behaviors. Now, the question of who's there. Well, first there are bacteria, and each person probably has 1,000 different species. Second are viruses. Now, most of these viruses are bacteriophages. That is, these are viruses that inhabit bacteria, and they alter the metabolism of the bacteria. There are fungi, mostly yeasts, about 15 species. There are archaea. These are primitive species that look like bacteria, but they produce methane gas. There are protozoa, which are one-celled animals, and helminths, or worms. And virtually everybody in the world harbors these. Now, taxonomy is the organization of taxa, which is the way that we understand and classify who's there. And it goes from kingdom as the broadest category, and phylum, next broadest, down to species and strain. There's tremendous complexity and diversity of the microbiome. For example, bacteria and archaea, which look identical under the microscope, actually exist in separate kingdoms. Plants and animals exist in separate kingdoms. And these distinctions are confirmed by DNA sequencing. Lactobacilli and bifidobacteria, which are generally thought of as probiotics, they're kind of lumped together, they actually are in separate phyla. Now, humans and eels share the same phylum, so that gives you an idea of the diversity of the organisms that are present. To complicate that matter, different strains of the same species may have divergent, even opposite effects. Bacteroides fragilis strains, for example, may prevent autism, but they may also promote colon cancer. When we add to this complexity the role of bacteriophages in altering bacterial metabolism, this is beyond quantum physics meets rocket science in its complexity. Now, in general, diversity and balance create a healthy ecosystem. So health is usually associated with a greater variety of taxa at all levels, and with a greater evenness in the richness of different taxa. Groups of unrelated taxa support one another by creating interdependent feeding networks called syntropic clusters. So basically, the microbiome consists of complex, dynamic microbial communities, and that complexity is increased by the activity of bacteriophages. Now, within this understanding, there are some bacterial species that are called keystone species. That is, they are major taxa that support other species to hold the whole system together. And one of the most important of these is Faecalibacterium prausnitzii. It is a major producer of butyrate. And butyrate, in addition to everything that it does to support the colon lining, to oppose inflammation, and its effects in the brain, also supports the growth of bifidobacteria, which ferment butyrate further, and the end product of that is acetate. Another keystone species, Akkermansia mucinifera, is receiving a lot of attention. So far, I haven't found a positive role for it in the setting of COVID-19. Now, Faecalibacterium prausnitzii is a major anti-inflammatory species. Chronic fatigue syndrome, ME, is associated with a reduction in F. prausnitzii and other species that are frequently described as anti-inflammatory, with decreased bacterial diversity and with an increase in pro-inflammatory species of the family called Enterobacteriaceae, which are basically gram-negative aerobic bacteria. That is, they grow well when oxygen is present. CFS-ME is also associated with elevation of blood markers that indicate increased penetration of the body by microbes, microbial translocation, like lipopolysaccharides or lipopolysaccharide binding protein and soluble CD14. So there's increased intestinal permeability, the so-called leaky gut, which is demonstrated in people with CFS-ME. Now, dysbiosis, a term that's been used in this symposium, basically represents an imbalance or instability among the many organisms of the microbiome that alters the ecosystem, creating undesirable effects on the health of the host. Stress can create dysbiosis. Now, mostly I'll be talking about the effects of dysbiosis on the brain, the central effects, but the brain itself can create a dysbiosis, and that, at least in part, is mediated by stress hormones. So adrenaline, which is produced by the sympathetic nervous system, and noradrenaline can selectively encourage the growth of some pathogenic bacteria. This has been demonstrated for E. coli. And adrenaline, in addition, can make bacteria like E. coli produce more toxins. So we know that people who are under stress are more prone to bacterial infection. This occurs, let's say, with surgery, for example. And most of the time we've thought about this as, well, stress is suppressing the immune system. What hasn't been fully appreciated is that the stress hormones directly stimulate the growth of some of these pathogenic bacteria. These toxic bacteria can break down the intestinal barrier, creating aggravating the leaky gut. This gut leakiness allows greater penetration of these toxins into your body. And there's evidence that stress management techniques, meditative practice, and yoga can help to control this chain of events. Now, inflammation and dysbiosis exist in a vicious cycle. And I think that's something that has not received the attention that it deserves, because you need to break that cycle for probiotics, for example, to be able to have a positive impact. What inflammation does is to increase the concentration of nitrates in the inflamed tissue. It does this by increasing the synthesis of nitric oxide, an important mediator of inflammation. A high nitrate environment changes bacterial growth patterns. Anti-inflammatory bacteria, like F. prausnitzii, tend to have their growth stunted in a high nitrate environment, whereas pro-inflammatory bacteria, especially E. coli and other gram-negative pathogens, their growth is stimulated. So their growth then creates more inflammation and maintains a high nitrate concentration. This is a feed-forward loop. From the perspective of the bacteria, it makes a lot of sense. But this is why inflammation needs to be addressed directly for dysbiosis to be controlled. Now, SARS-CoV-2 in the gut creates dysbiosis. One mechanism by which it does this is independent of inflammation. SARS-CoV-2, as is well known, enters human cells through attachment to the vital enzyme ACE2. When it attaches to ACE2, its entry into the cells decreases ACE2 activity. This has been demonstrated in blood vessels, in the laboratory. It's been demonstrated in numerous tissues. Now, ACE2 has a particular function in the gut that is unique. Aside from its role as a vital enzyme, it acts as a chaperone for the transport of certain amino acids into the body. So the loss of ACE2 in the small intestine, that is an enteric ACE2 deficit, impairs absorption, and particularly affected more than any other amino acid is tryptophan. The loss of tryptophan depletes serotonin in the gut, and the loss of serotonin diminishes the synthesis of these important proteins called defensins that help to regulate the composition of the gut microbiome, prevent viral and other types of infections. So the effect of the loss of ACE2 by SARS-CoV-2 infection is to produce a gut bacterial dysbiosis. Now, there is an impact of COVID-19 on the gut microbiome that has been measured and that has been shown to persist for months. This includes decrease in bacterial diversity and an increase in the relative numbers of pro-inflammatory species. There's also an effect on the fungal part of the microbiome. There's an increase in fungal richness. Candida albicans, Candida auris, Aspergillus flavus have all been shown to increase. And Candida auris has received a lot of attention lately because it is a drug-resistant, very dangerous fungus that is killing people in hospitals. Now, researchers at University of Massachusetts found that there was one particular species that, when increased, was associated with increased mortality. And this is Enterococcus fecalis. Now, Enterococcus fecalis is sometimes used in certain probiotics. It is an immune stimulatory species. It is a potent inducer of gamma interferon, and gamma interferon plays a role in driving the cytokine storm of severe COVID. Now, the keystone depleted species is Faecalibacterium prausnitzii. That is lost in the course of COVID-19. The mechanism by which it's lost isn't clear, and so I'm fascinated by Dr. Bronia's work, which explains a mechanism that I had not anticipated. Now, another study looking at F. prausnitzii found that the gut microbiome at disease onset predicts the development of PASC. And the gut microbiome of patients who develop PASC is characterized by lower levels of F. prausnitzii when they first get sick, and higher levels of a couple of other species. Ruminococcus navis in particular can be very inflammatory. The largest inverse correlation between the development at PASC at 6 months and the microbiome at the time of disease onset was with these butyrate bacterial species, F. prausnitzii, and also a bifidobacterium species called Pseudocatenulatum. Another issue that has come up within the setting of COVID-19 is whether persisting viral infection can contribute to PASC. There are a number of studies. In the interest of time, I won't go into all of them here, but there are a number of studies indicating that it's likely that there is continuing viral infection in people who develop PASC, and this has been looked at in people with neurologic symptoms. Program that I've put together is a high-fiber, polyphenol-rich diet, which increases the growth of F. prausnitzii. Adherence to a Mediterranean diet increases F. prausnitzii. Prebiotic oligosaccharides may as well. We can raise the question, and maybe we'll try to answer this, whether that's actually desirable given Dr. Bronia's work. There are several probiotics and prebiotics that increase the growth of F. prausnitzii. And I just, I'm going to pass over the impact of the gut microbiome on cytotoxic T lymphocytes, unless you want to give me an extra minute or two, Philip.
Yes, go ahead. I think that what
Okay.
we'll do is we'll kind of deduct it from our discussion. Okay. Yes, go ahead with that.
Okay, yeah, so it's become very clear that the outcome of COVID-19 is very much impacted by T lymphocytes, in particular cytotoxic T lymphocytes that kill virally infected cells. They play an important role in recovery from viral infection. They're critical for vaccine-induced protection against this virus. And I think there's been way too much importance attributed to neutralizing antibodies. It's really the T effector memory cells that are cytotoxic lymphocytes that are important for protection. Now, lactobacillus consumption through yogurt, fermented foods, or as a supplement increases the number and activity of cytotoxic T lymphocytes. Lactobacillus plantarum may be particularly important. And the question of whether do you want to give probiotics to people with COVID-19 because of the bacteriophage action of the virus, I think that's a very important question. I've only seen one study that might suggest an answer to that, a study from Spain, and I don't have a slide on it, I hadn't prepared for it, in which a fixed probiotic combination that included a strain of Lactobacillus plantarum was given to people who were acutely ill versus placebo. And the people who got the probiotic had a much better, they had a shorter course and a better outcome than people who did not, who received the placebo that did not contain the probiotics. So at least when it comes to lactobacilli, there's evidence of benefit. There are a couple of other things that may enhance T effector memory cells, which are a specific type of cytotoxic T cells. These cells thrive on fatty acid oxidation, the availability of butyrate, and the presence of lactobacilli. So a high fiber, adequate fat, low sugar diet with fermented foods may be helpful. Carnitine has been shown to help their development and may be a worthwhile dietary supplement. Now, just coming back and trying to close the circle on bacteriophages. There is some work looking at dietary factors that impact bacteriophage activity. And I think this is an important area for research. Polyphenols, especially bioflavonoids, disable or kill bacteriophages. And the mechanism is inducing structural damage to the capsid, inhibiting their infectivity and their activity. Oregano oil has been shown to suppress multiple types of prophages, preventing their cellular entry and impact on bacteria. In contrast, the natural sweetener stevia has been shown to promote multiple species of phage to increase in activity and increase their impact on bacteria. And stevia is very widely used by people who are trying to avoid sugar. It would be interesting to look at the relationship between stevia use and the outcome of COVID-19. Thank you for your attention and the extra time.
Excellent. Thank you very, very much. This is very interesting to me. So I will not start with my questions. I will let Valentina go first. And we'll probably have to shorten this period, but when we go to the open forum, we can expand on any further questions when we bring everyone in. So go ahead, Valentina.
So thank you very much. First of all, I would like to thank Dr. Galland for his huge contribution to the knowledge at Long COVID Foundation, where Dr. Galland has shared many informational presentations. So I would like to take this opportunity and invite our listeners who suffer with Long COVID to join Long COVID Foundation's channel. And I believe you can find the link under this video. So for now, I would like to proceed to some questions to understand the importance of gut microbiome, because people who suffer with long COVID, they are completely confused from which angle to start treatments. So what is your view, knowing that we have seen Dr. Bronia presentation and discussion about the combined treatment of antibiotics together with probiotics. So there is lots of information that antibiotics don't work on viruses. So it's a little bit confusing for people maybe to understand the real benefit of antibiotics during acute illness. And then in terms of the effect of antibiotics might do on gut microbiome, how difficult it is to choose the right probiotics to restore the gut microbiome?
Yeah, these are really good questions, and they're not easy to answer, Valentina.
As
And
always.
also, you know, Dr. Brogna's work was called to my attention by you about 2 weeks ago, and I just haven't had a chance to really figure out how I would work that into the protocols that I'm using with patients. I share his belief that widespread antibiotic use is not the answer. And I think that there are a lot of risks there. Over the past 2 years since the onset of the pandemic, there have been practitioners who have advocated the use of antibiotics. Azithromycin is frequently administered, sometimes doxycycline, and sometimes with other substances. I've only seen one study which was kind of a retrospective analysis as to whether azithromycin seemed to improve outcome with acute COVID, and it did not. So in that study, so I'm not sure where I would go with antibiotics. I like the idea of rifaximin because it's not systemically absorbed, but it's ridiculously overpriced. So I don't think that's a solution either. I would like to, I think the future there is going to be found with bioflavonoids and determining what are the bioflavonoids that are most able to disable, the bacteriophage activity of the virus. There is a probiotic. When it comes to probiotics, it's kind of hard to know what's the ideal probiotic. What are you trying to accomplish? Originally, when I saw the data on F-Prausnitzi and butyrate production being inversely associated with outcome of COVID-19, I thought of this as being a manifestation of previous diet and lifestyle. That is, healthier people have more F-Prausnitzi and they have greater butyrate production. So I thought we were just looking at an epiphenomenon. Yeah, the reason that these people are doing worse is because they were not healthy to begin with and they had an unhealthy microbiome to begin with. Dr. Brogna's work suggests that maybe the infection itself is really the main driver for this kind of dysbiosis. And so the question is, well, do we want to build up F-Prausnitzi or don't we? Maybe we want to do something else. Their lactobacilli, I think, are more likely to have a positive effect, especially plantarum. And so the use of fermented foods or lactobacillus plantarum supplements is the direction that I would like to look in at this point. There are this class of organisms called soil-based organisms. The genus is Bacillus, Bacillus clausii, which Carlo mentioned is one species within that genus. There are others. Bacillus subtilis has gotten a lot of attention, and there's a particular strain of that that was originated actually in Ukraine and is still available, that generates alpha interferon, which kills this virus. So I've used that particular product with patients and sometimes seen really excellent results, especially when they have GI symptoms that are part of COVID-19. And I attributed that to the impact of the alpha interferon being produced in the gut and killing the virus.
Thank you so much. If I may, Philip, have a
Go
couple
ahead.
of more
Yes.
questions. So you said some bacteria can produce toxins, and what would be the best available protocol to detox intestines from your experience? And would it make sense to have some kind of zeolites or charcoal or humic acid?
Well, all of those may be possible. I just haven't been using them. I hadn't been thinking about long COVID as really a manifestation of bacterial toxicity, but rather as the absence of butyrate producing bacteria. So my protocols have been finding ways to enhance the growth of healthy bacteria, control inflammation, which is essential, usually with substances like curcumin and resveratrol, quercetin. And the important thing in dealing with natural products like this is there can be big problems when you're trying to go from laboratory to clinical practice because they're not that well absorbed. If we're dealing with the gut, systemic absorption and systemic effects are not so important because you really want the effects in the gut. So if something like quercetin is not well absorbed, but it stays in the gut, well, maybe that's where you want it.
Thank you. Thank you. And the last question from me would be, as I usually do at the Foundation's channel, we take some questions from our patients who suffer with long COVID and share their best experiences about the disease. So there is a question from a person who suffers with mast cell activation and dysbiosis. So if there is a dysbiosis in the gut and deficit of some bacteria, for example, Enterococcus species, so which are in kind of histamine builder. So how smart it would be to take probiotics which grow histamine building bacteria, especially when you have mast cell activation and you need to take probiotics to decrease histamine. Very practical question. Maybe
yeah, I think it is. In many of my, well, first of all, I think it's important to understand that mast cell activation and histamine intolerance, although they often overlap, are not exactly the same. That is, there are people who have mast cell activation syndromes who are not particularly histamine intolerant. And there are other chemicals that the mast cells are releasing that are making them sick. And then there are people whose problems are related to histamine intolerance. They have difficulty breaking down histamine. This might be due to a copper deficiency or a methylation problem. And they may not have a general mast cell problem. There are probiotic bacteria that either produce histamine or break down histamine. And when I'm working with patients with mast cell problems or histamine intolerance, I will generally recommend probiotics that are histamine, that break down histamine. And they're specific, and this is strain specific. This is not even species specific.
Okay, thank you so much. That's all for me.
I will leave my point to one because we're running out of time, but I'll ask some questions afterwards. But I'll just say, Leo, you are right on my area of research with what you've said, especially with the tryptophan bit. This is something I am focused on, the immune-mediated mechanisms in COVID-19. I'm going to make a statement, and I'd love to hear your thoughts on it. I could predict the patients who were going to get long COVID before they've got COVID. And it's really because of three symptoms: diarrhea, bloating, and constipation. If they had any of those symptoms before they had COVID, I found that they had high risk clinically of getting long COVID. And it tended to mirror inflammatory bowel disease, where the highest percentage of patients who got long COVID had IBD. So I eventually came to a conclusion that I'm currently looking into, which is that the gut microbiome is altered in IBS and IBD. And actually, when you look at celiac disease, it also has an altered gut microbiome. Is it feasible that this is primarily because if there is a previous allergy in the gut that is producing IgA, it's damaging the microbiome and then making people more vulnerable for gut-related symptoms?
Well, that certainly could be the case, Philip. And if the other thing is that allergic reactions in the gut and inflammation in the gut increase its leakiness. They increase the permeability. So then you get increased absorption of things like the lipopolysaccharides. What happens with COVID-19 and with long COVID is really, really complex. There are multiple systems that interact with one another. And I don't think we're going to find one path that ties everything together.
Yes, yes, that's a good, but that's what I'm looking for. I'm looking for the Holy Grail.
Well, I sent Valentina a scheme that I put together a diagram called the Web of Long COVID, which was kind of the best I could come to trying to, in a visual way, express the interrelationships between different systems, and the symptoms are not in there, but organs and systems and functions are, and the way a lot of this derives from the fundamental effects of the virus, starting with the destruction of ACE2 on viral entry.
That's really true. And yes, I would like to say that the complexity of long COVID, as we see now, it's even more complex. And I think it's very important that we are all here tonight and we are bridging some gaps between the brain and microbiome. And we have this opportunity to discuss and maybe rethink our future strategies on treatments. So that's my big thank you to all of you who are here today. So I'll let you continue, Philip.
Wonderful. So what we're doing now is we're kind of opening up the discussion. And so for people who are listening, this is where we just now talk with each other. We've heard some fascinating presentations. And what I'm likely to do is that this part of the discussion will be for 30 minutes. I will then do a formal close of the conference. So I'll do the outro, but I'll ask anybody who can stay further for us to continue to talk afterwards. And that's for the audience. And we'll take some questions and so on if I can, if you put them in. So anybody wants to go first? I just wanted to say Carla, the work that you have done with regards to the bacterial involvement in terms of infection is a game changer. I can, that's all I can say, is that it adds so much to the understanding of the disease. What made you think of doing that?
The toxins. So at the beginning, we didn't realize that bacteria was a cofactor in this disease. And when we found the toxins in the plasma of COVID-19 patients, and we observed the mass spectrometry of each toxin, we noted the variation of amino acidic. Every time the conotoxin was presented more time in the same patients with every time an amino acidic variation. Every time, 10 times, 20 times, 30 times. So when I look at this amino acidic variation, I remember of polycystic mechanism of bacteria. And I asked me, why these toxins have variation so frequent? And maybe the bacteria produce them. So we performed the culture to found the toxin production in the fecal bacterial culture after 30 days. And the control of RNA increase was only an internal control. We don't think, we didn't think the bacteriophage mechanism. It was a very è stato un caso, come dire? Was a case to found this observation. Yes.
Yes, chance observation. Wow, tremendous. And Mike, I'm just curious from your perspective with regards to the neural symptoms, because one of the arguments has always been that the virus is continuing to replicate in brain tissue, and this is what's driving the symptoms. Any thoughts with how what you are looking at from a neural perspective could tie in with what Carlo is seeing?
I mean, if the virus had penetrated into brain and was replicating, that certainly could be an explanation. But I don't think that has to be the case for there to be neural consequences at all. So peripheral inflammation can drive excitation towards the brain. So, vagus nerve to brainstem, and then it triggers what's called a mirror response on the brain side of the blood-brain barrier, where glia activate. And the activated glia kind of act like amplifiers. So that can be on stress-related circuitry, triggering depression. If someone's got some vulnerability to different forms of psychopathology, that could, drive those problems. So I don't think it has to be the case that it's virus in brain. That certainly would be bad news if that were happening. But I don't think it has to be for there to be really profound neurological consequences if inflammation or problems with vasculature were driving it.
Great. Joachim, go ahead. You wanted a question? You're muted at the moment.
So sorry. One question to Mike, a quick one. We are just finishing trials in animal trials in India on our phenolic compounds, the mix of these flavonoids and compounds on M1, M2 state macrophages. And we have seen already the results that it is helping to stop this activation. Would that also theoretically work on glia?
Yeah, I mean, microglia are the resident macrophages of the brain. So during development, macrophages form in the yolk sac, and then during development, they sort of move to different areas of the body and take up residency. So there's resident macrophages all over the body, in the endometriosal tissue all over. And in the brain, the resident macrophages are just the microglia. So something that works on microglia, if it has brain penetrance, would probably affect the microglia as well.
I'll share that with you then, and then we have a look at it. Thank you.
Great. Manan, go ahead.
Yeah. I was so much tempted, when Carlos was presenting, that I was wishing that if I would be the panelist there, to ask this question, and then we will have a panel discussion. So the question is for Carlos, Dr. Carlos, thank you very much for your very nice presentation. What I was wondering is that there are two bits to my question. Number one is that if the bacteria in the gut are actually harboring the virus, do you think that the toxins that they are releasing could escape to portal vein and liver and into the blood, and they could be contributing to the signs and symptoms of long COVID? What are your thoughts on that?
Ma che cosa ha detto? Ok.
Ha detto, non pensi che gli elementi che stanno nella parte cerebrale possano uscire fuoriuscire ed andare in altre parti del corpo?
Oh, yes, of course. Thank you for the question. Of course, it is possible, everything. We don't exclude nothing, and we much more study to understand the mechanism. We are providing we are make some experiment on the midges, on the midges, and we are waiting for the results. And we also are performing experiment on neuronal cell with the toxin. And but in this moment we have not the results, so I can't explain everything in the right way. But everything is possible. We don't exclude the double mechanism of SARS-CoV-2. SARS-CoV-2 obviously attacks the human cell and the epithelial cell, but in our tests, in our experiments, it also attacks the bacteria. So the toxins maybe can be produced also by human cell. We can exclude everything, but we must perform more experiments. So we are at the beginning of this new way, and so it's premature to definitively. It's too early to say something on or not.
Do you think that if we develop a strategy, in which we can make keep the bacteria, not kill it, but make it vomit the virus, and then give some oral antivirals, as Dr. Leo mentioned that sometimes the antibiotic or antiviral which has less absorption from the gut is a good news, So you make the bacteria vomit, and then you give that antiviral that's not absorbed very good from the gut, that can locally kill the virus. So I'm just thinking loud, because destruction of the microbe will actually change the what you call as normal homeostasis of the body. Do you think that it would be a viable therapy?
Yes, it is possible. I can't exclude also this observation. And so we have focused attention on one aspect, but we need, in reality, we need more investigated the process.
Thank you.
Can I, just before Shankara, so Shankara is going to make, and then Leo, but I'll just make a quick point to Manan. One of the things that I have been looking at carefully that can answer some of the problems that we're seeing with the microbiome issue is literally the fact that many people have a buildup of fecal matter with abnormal bacteria. If you just completely clear their bowels, almost as if you're doing a colonoscopy, you are likely to remove quite a bit and allow a rebuilding of the gut microbiome as a simple mechanism to start it. Okay, Shankara, go ahead, your question.
Philip, more of a comment. We're noticing a variety of mechanisms at play. With COVID infection, we know that we develop a robust immune response to it. And of course, we know that in certain patients, there is an inappropriate immune-mediated response that can be very destructive. And of course, now we have the evidence that Carlo has presented. We were aware of positive PCR fecal swabs, but from his work, we're seeing that it does infect bacteria in the gut itself, which causes two things, or results in two things. One is that the bacteria can cause replication of this virus and cause that persistence of virus in the gut itself. And other bacteria can produce toxins. So we've got many mechanisms at play. So we can have an individual who develops robust immunity and now does not get infected with the virus, but harbors the virus in their gut. And that can, in those patients that are actually allergic, expose them to a constant allergen and result in some of the sequelae we see. So you've got an allergenic propensity, you've got an infective propensity, you also have toxins produced. So I think the mechanisms at play are multifold and dependent on the patient itself.
Yes, I agree, yeah.
Leo, go ahead.
Carlo, I have a question for you about the impact of the toxins that you're looking at on acetylcholine. It seemed as if some of these were cholinesterase inhibitors.
Wait a moment, please.
Le tossine che tu hai scoperto,
Sì.
che tu stai trattando, possono essere considerate anche come inibitori del Covid.
Okay, yes, it's a good observation, yes. I think that the bacteria produce the toxin. I show you a slide, the number 23, where in one culture, where we remove SARS-CoV-2 after that they touched the bacteria continue to produce the toxin, and the viral load of SARS-CoV-2 decrease. So maybe the toxin are a mechanism in our mind, a mechanism to yes. No, the next picture. No, the first. Also, okay. So the sample C is the sample where SARS-CoV-2. This sample C is the sample where the supernatant of SARS-CoV-2 was removed, but the level toxin is the same of the sample A and the sample B. In the sample C, the viral load decreased very quickly. So, but not at the point 0. It continued to be still a little present. So, in our opinion, but we must more study, in our opinion, the toxins are a mechanism to destroy the virus.
So you mean it's a response by the bacteria to try and control the bacteriophage?
Yes.
So then the toxin, this will tie in, Leo, with what you are seeing, because you are noticing particular patterns of bacterial growth. So dependent on which bacteria are in I guess, in the primary numbers will determine potentially which toxins are produced and therefore what effects it could have on the body. So in effect, we're looking at a microbiome disease.
Yes, I think that it's very likely that the impact of the bacteriophage on the specific bacterial species may depend on the characteristics of the bacterium just as much as it does on the characteristics of the virus. Has anyone ever seen or have any experience with a virus that can act as a bacteriophage and also infect humans and other mammals?
I think I was looking at that as well, and I think one of the beta coronaviruses could do something similar. So it's not impossible, and it does happen, but not very often. This is certainly an unusual characteristic.
Yeah.
Joachim, you're going to say something? You're muted at the moment, Joachim.
Carlos has documented that well in his presentation. You can look we will share the slides anyhow between each other. So Carlos has documented that exactly the same mechanism that we see now has already been observed with other viruses. So it's not a completely new thing.
Absolutely. And it raises a question then I guess this is the point with the vagus nerve with regards to Mike, because as you pointed out, the vagus nerve is so well spread throughout the GI system. Could this be a mechanism for vagal stimulation, this kind of toxin impact in the gut, dysbiosis?
Yeah, and if there's gut dysbiosis that's causing inflammatory mediators to be released by the human cells, then that will be detected by the chemoreceptors on the vagus nerve afferent terminal. So, any inflammation in the trunk organs, lungs, gut, etc., would be detected and might trigger the feeling of being sick.
Mm. Yeah, go ahead, Leo.
Yeah, Mike, have you looked at low-dose naltrexone and its effects on microglial stimulation?
Yeah, it's a really interesting question because it does seem like, low-dose naltrexone is a really interesting question because you think of it as being sort of like a mu opioid effects, but it must have effects beyond that. We just need to study it better. It seems like it has some effects on natural killer cell function. There is some argument that it is a modulator of glial cells without being necessarily immunosuppressive, which I don't think you want. I wish people would study that more. It would be really good to know more about.
Are there other substances that you know of that might work as modulators of glial cell function? Curcumin, for example, that are not immune suppressive, but would control the glia.
Yeah, there's actually a lot of different things that affect glia at one point or the other. And, I always think it's really important to note that, if there's a way to address a root cause, you want to do that first before you go to try and affecting the glia. But given that they're, that they can be primed and have problems there as well, it's something that you can address. But yes, there are, like, so for example, some even antidepressants. Antidepressant is just a marketing term, right? There's that's sort of what it's been tested for. But depression can be driven by glial activation as well. So there's a lot of different drugs. There's AV411 that's used in Japan as an anti-asthma drug. Amitriptyline is used as a glial inhibitor in some cases. There's a whole bunch. If you want to look into it, there's a good review article. The lead author on that was Peter Grace, Nature Reviews Neuroscience, maybe 2017, called Pathological Pain in the Neuroimmune Interface. People are studying, a lot of pathological pain is driven by glia pumping out neuroexcitation as well. So there's a lot of different things that are being studied.
I wanted to specifically bring in Valentina here. As we talk about some of these very complex topics with the microbiome and the neurotoxicity, what is going through the heads of the patients who have got long COVID and just want to get better?
Well, the most important thing is get treatment now. And I think it's very fair to say that 2 years on, we still don't have anything that would particularly tackle the root cause of the disease. We still are trying to understand what is happening with all these multisystemic symptoms, and people are really unhappy about the medical care they're getting. So I think this is a very unique opportunity for people to watch these videos and learn from experts who are discussing root causes and analyze lots of research papers for them to bring to their practitioners and spread the awareness about the real causes and the importance of these findings that you are discussing here today. That's number one. And number two, I think, is we are now at the stage to discuss antivirals and antibiotics, and people with long COVID cannot get the supply of antivirals because they're not within the group of disability. So they're not characterized as people in need for antivirals, and it's a huge issue for them now to basically raise awareness that long COVID is classified as a disability because many people are in beds for years with no treatment. So antivirals is very important for them now. And I just wanted to ask all of you here, what is your opinion on antivirals? Is it really the number one thing that long COVID sufferers need to take, or are there any other solutions or nutraceuticals that potentially might do the job as good as antivirals?
I'll start off with that question, Valentina, because it's a very good question, and I've heard it before with the antivirals. The only thing that I would say is that one of the difficulties both for antivirals and antibiotics is about how much of it remains in the gut, because from what Carlo has demonstrated, the virus is actually replicating in fecal matter. So it's not in the systemic system, it's in the gut system. And therefore, if the antiviral is not able to concentrate well enough, is it even able to penetrate the bacteria to impact on the virus? There's so many questions about it that my suspicion would be that whilst in theory it sounds good, it may not necessarily work out in clinical practice. Any other thoughts? Manan, go ahead.
No, okay.
So I would like to start from where Dr. Leo left us, that sometimes a decreased absorption of an antibiotic is good news, So if you are targeting gut microbes, and you come across a molecule which is not absorbed very efficiently through the gut, it might help reduce the viral load. To answer, connecting one into it, is that we first have to establish that persistence of the virus is the line one cause in long COVID. If that research gets accelerated, then there are several branches, like once the persistence of the virus is there, then you can branch it into 20 to 25, what you call as subdivisions, as Dr. Leo was mentioning, that he has got a figure, cardiovascular system, renal system, pancreatic system, and then it branches out. So where it actually comes, where is the epicenter? That's the $1 million question. You understand, Dr. Philip, you pointed out in the previous conversation that we had, that you want to go for that magic pill, isn't it? The single cause, you just catch hold of it. I would urge my community and all the persons who are involved in research to search for that pill. I mean, see whether the reactivation of the virus, it's hiding places like Carlos mentioned. You can't imagine the crypts of the small intestine where it could go and it can hide itself, and it keeps on emerging through the portal vein. We always will have this problem. So we have to address something, which actually finds that that research niche, of that persistence of the virus or any other cause. I'm open. I mean, data could prove it, but then we have to find the first ball, which splits into 22, 25 balls.
Yeah. Joachim, you wanted to make a point?
Very quickly on the antiviral properties of natural compounds, phenolic compounds. So if you ingest flavonoids or other phenolic compounds like curcumin, quercetin, they're all in the same class, more or less. They will be metabolized in the gut. That is the first thing that happens to them. They're primed a bit by the saliva, but once they go into the gut, they'll be metabolized. And many of them have very, very strong 3CL inhibition properties, to say something, against SARS-CoV-2. And so then you will have the maximum availability, so to speak, ground zero in the guts for these natural compounds. So very likely, and I will go and fly myself to Carlo and we'll try that.
Yeah. Listen, what we're going to do now is that we're going to formally close out. I don't know. I know Mike is not feeling well, so he may not be able to stay with us, but we're going to formally close. Anyone who can stay afterwards, please do, and we can continue to talk. Before we close out, I just wanted an opportunity to ask Valentina first and then Joachim, just to share a thought with regards to the vision or the idea of the Long COVID Coalition. What do you think, or what do you see where this could be? Go ahead, Valentina.
Thank you. So, first of all, we are facing so many challenges, and we see that medics and scientists are very much into a very small direction of the research field, and we are facing the disease, which is novel and needs collaboration between all medics and scientists in order to tackle it. And we already know that by many research papers and many interviews in the past, we agree now that this disease needs to be tackled by many experts in the field. Now, with the coalition, we think that it is wise to bring all these experts on the table and just basically analyze these issues and share thoughts on one or another research that basically would demonstrate today that we find some great experts and help them to share research findings and collaborate together with medics and potentially discuss treatments. Because what we do, we bring patient-centered approach. So we want to help patients suffering with long COVID and identify solutions that would tackle these main root causes of the disease. And if there are people in the audience who have got some thoughts on how to tackle, have got experience on natural solutions that would help us build these protocols to help long COVID sufferers, then please do get in touch with us, and we want to collaborate. We want long COVID advocacy groups to help us know the disease better, to listen to those patients who suffer with long COVID globally, as we do at the foundation. So everyone is welcome to collaborate within the coalition, but we all want to help people, not commercialize things, but we want to bring expertise together so that we can tackle this disease collaboratively, not from a single perspective.
Excellent. And I'll ask Joachim to just give a quick thought as well.
Yeah, I have very good news for all panelists today. So you're automatically now part of the Long COVID Coalition. I will share every new developments. And Philip, can you be so nice and share very quickly the slide I sent you with the wrap-up that we tried to do before this meeting. And so this wrap-up is now not, of course, finished. It is halfway through, and I will share that with everybody that was participating today to give their thoughts, because in order to be beneficial for each other in the collaboration and for the patients, we need to have the correct description of the pathway and the problem, the diagnostic tools, and the possible treatments. So if any of you is interested and have a good look at this, then fill in whatever you think is appropriate as a treatment or diagnostic tool, or if there needs to be more added. I try to be throughout as far as I could have any prognosis how this meeting will go. So yeah, thank you for all new members of the Long COVID Coalition, and we will keep you informed, and the website will be up in the next days, and we will put each of yours presentation there if that isn't okay with you. And let's make this a living collaboration so that it's not only just meeting and share some thoughts, but we have in between the possibility to connect with each other, share knowledge, share ideas and experiences in order to really make that a collaboration that is alive. Thank you, everybody, for participating.
Wonderful, thank you. So I'm going to do our closing video for the audience. If you want to stay with us while we chat a little bit longer afterwards, please do. If any of the speakers or panelists can stay with us, that would be wonderful, but we understand if everybody has to go. So again, thank you very, very much, everyone, for this wonderful, inspiring, stimulating, educational conversation. And it has certainly opened my eyes to different routes and pathways that we have in front of us for long COVID. Thank you again, everyone, and thank you, everyone, for staying with us. And if you want to stay with us longer, you certainly can. Have a great evening, everyone.
Mike, you take care of your recovery.
Bye-bye.
Bye.